So thanks, Lutz. Yes, so my name is Efstathios Diamantopoulos. I am a professor at the University of Bayreuth of soil physics, and today I'm going to present, I would say, an incomplete, it's still not complete, work that we are doing at the University of Bayreuth, dealing with a modeling framework for temporal changes in soil structure and their effect on soil hydraulic properties. So it will not only be, I will not only talk about modeling, but I will also present some data that we are collecting. And actually what you see here with these 4 pictures, at the bottom of my first slide is how the field was at the time where we did some sampling. So I'm going to present some data, and I just want you to remember those pictures. This was exactly the day of the sampling, so of the field. Okay, I think for introduction, I don't think I have to stay long here. Nick did an excellent job, but if you think about the soil plant atmosphere system, and if you see the boundary conditions, we have changes that are taking place from seconds to centuries. For example, precipitation, temperature is changing, the wind is changing, we are applying different crops to the field annually, we change our crop rotation, input of matter changes, we are humans also changing the system by applying management operation like compacting the soil or plowing the soil. We have also the fauna, which is very dynamic. And I just did this slide just to highlight that there is a paradox here. Everything around the system changes, but in our conceptual understanding, in our, let's say, state-of-the-art models, we considered soil as fixed. And I'm saying that with respect to soil structure and soil hydraulic properties, which is our interest here. This is another slide from Nick, so I'm not going to stay very long here, but I think I will just highlight that we know that soil structure is changing, is temporal dynamic, and we have changes that are taking place in the short term, in, let's say, in the season, but also in the long term. So here I have some examples: tillage, freezing and thawing, shrinking, swelling, biological activity, earthworm, root production, etc. But we also have some long-term changes, like the climate is changing, the crops and the input of carbon is changing, the land use may change. So we know that these affect the soil structure. We know that for many, many years. The question is, does it matter? Should we account for those processes, for these dynamics in our models? And actually, this is a question, still a question for us. And in order to do that, we would like to do that a little bit systematically. Here I have some examples, like from our group, from research in our group, that indicate that soil structure should matter. I will not stay it very long, but I would say that we all know that preferential water flow exists. This means that water can flow through the big voids of the soil and bypassing the matrix. And if there is some chemical diluted in that water, then we can have fast transport of chemicals. This is very well known, but also maybe something that is not so very well known is that soil structure can also affect bare soil evaporation. For example, there is a saying among the German farmers that if you plow your soil once, you may save more water, you may have to irrigate less during the summer. And this is something we try to, for example, to evaluate here in a study in a group where we took, let's say, different cores from plowed soil and the non-plowed soil. We took X-ray computer tomography, and then we simulated let's say we resolved the 3D structure, and then we simulated bare soil evaporation. And what you can see very nicely with these simple simulations is that for the plowed soil here on the top, the first millimeter of the soil dries out much faster compared with this more continuous matrix in the grassland. And since this part of the first millimeters here drying out, what we have at the end that this topsoil here act as an isolated layer, and in that way we have a reduction of the bare soil evaporation, and at the end we can save more water at the bottom. So soil structure is affecting also bare soil evaporation. When we go to soil reactivity, these are also some results from some synthetic simulations that we have conducted here with Christopher Henry from GEUS from Denmark. And actually, it's a very simple exercise. We have a simulation at the plot scale where we have some kind of heterogeneity. And the question that we try to answer is, does this heterogeneity affect soil reactivity? And what we found very, very clear is that soil heterogeneity, but also water fluxes affect soil reactivity, effective soil reactivity. And this means that the assumption that we do in our models to describe reactivity at the numerical cell with one coefficient is very, very weak, at least for a system where we know the whole truth. And if, I mean, it's an idea, if we would like to come up with more robust reaction models, we should account actually for this kind of heterogeneity, physical heterogeneity. And the last thing had to do with biological activity. This is a study from Leuter et al., where they just tried to relate it biological activity with structural indicator. This is happening at smaller scale. And what they showed very nice with this correlation matrix is that structural indicators are correlated with the biological activity. And more or less we had a surface density. This is the surface area, let's say, of the structural porosity, but also the distance from the matrix to the nearest structural pore are correlated with the biological activity, indicating that there is a link between soil structure and biological activity. So these are some internal work, but if you think about it, I just named some other processes that I'm expecting to be affected by soil structure, like gas transport, aeration of the soil, root growth, heat transport, mechanical processes. And maybe after, in the question round, maybe we can think other processes. So all our experience, all our understanding, all our prior knowledge indicate that there should be an effect. And this is actually what we try to investigate here in Bayreuth. And what I'm going to present is the work of two PhD students, Mathilde Nielsen, who is doing the experimental investigation. Actually, first of all, we want to generate good quality data and answer the question, do soil hydraulic properties change with time? And then Daria started 1 year later than Mathilde, and she is working with the model that I'm going to present to you. Okay, so to quantify if soil hydraulic properties change with time, we had to work with a long-term experiment, and we worked with a 30 years experiment in Puch, Bavaria. So actually, they are testing 4 different management treatments for 30 years in a randomized block experiments that you see here. The good thing is that the soil texture is identical. Oh, sorry, is identical for all the plots, and they have 4 different management treatments: direct seeding, extensive mulching, intensive mulching, and conventional plowing. We went there many times in the last 2 years, and we collected samples from the topsoil and from the subsoil. I'm going to present today more data from the topsoil. There is, of course, a crop rotation and management operations, which are quite important for the soil structure. So as I said, I'm going to talk more about the topsoil, but I want also to highlight here that, as we discussed also before, in these 30 years, they have some measurements also, not of the soil hydraulic properties, but at least they have measured the bulk density, the water content at field capacity, the water content are wilting point. So my hope here is that we will put everything in a nice database and we will be able also to calibrate the model that I'm going to present through this 30 years data. Okay. Okay, so to measure the soil hydraulic properties, we had to develop a new experimental protocol. Yes, I know we think that soil hydraulic properties is quite easy to measure, but still for this kind of research question, we had to really develop a new experimental workflow. Actually, what we did, we did some 4 experiments in series. So first we saturated the sample, then we measured the saturated hydraulic conductivity. Then we use this multi-step flux device to measure near saturation, unsaturated hydraulic conductivity. Since we have drained a little bit the sample, we resaturate them, and then we continue with the HYPROB and the WP4C method. And if you see some results of this experimental workflow, so at the top here we have the water content, and on the bottom here we have the conductivity function, and you see more or less with red points, you see the measurements of this multi-step flux device. I don't know if everybody you're aware with that, but actually what we have a soil sample. We apply an irrigation flux on the top. We measure with the tensiometer the suction near the top of the column, and we apply that automatically as a suction at the bottom of the column here. And in that way, we drive the system very fast. We bring the system to unit gradient conditions, meaning that there are no pressure head difference among the column. The only force that is driving water flow is gravity. You can think of it as we have an infinite soil column, and this is the closest thing we can do to measure this unsaturated hydraulic conductivity for field conditions. And as I said, with the red point, I have highlighted those MSF data because they are quite important when we investigate soil structure. And we first tested the method for a sandy soil, a model soil with a very well developed, let's say, very well defined air entry. And this was mainly to see if we actually, the MSF data fit with the KSAT, but also with the HYPROB data. And I hope you agree with me that it's doing a very good job. But I would like to focus on these silty loam 1 and silty loam 2 samples, where in the conductivity curves, you see that we have some samples that the MSF data follow very nicely the hydraulic conductivity, the saturated hydraulic conductivity here, and this kind of Mualem type of model can describe that very well. However, we have also plenty of samples that show this kind of behavior. And here, if we omit the MSF data from the fitting, you see that we can do like a 2 orders of magnitude error. Which one now it's better for the simulation is something that we can discuss later. Okay, so here are some data, and again, it's not the complete data series, it's just only for the first 3 points. And you see here the water retention curve for the direct seeding and the water retention curve for the conventional plowing. And let's start a little bit from the dry range. You see that for both treatment, but also for the different time points, it seems that this part of the curve is not affected so much, and this makes sense in my understanding, because these are data that are more or less a function of the soil texture, and since we chose a field that the soil texture is very similar, so we don't see a big difference. However, what we see a difference for the HYPROB data in the wet range, let's say from PF-1 to PF3 at least, but we also see a difference between the time points and between the different treatments. For example, you see here that the slope of the retention curve for the conventional plowing is more steep, indicating different pore size distribution. So these changes, we can translate that in the pore size distribution. Here, I also put that, I think I just asked Matilde today to give me that just to see all the 6 points that we have collected until now. This is the plant available water content, and you see this, that this changes really a lot between the different times point, and this difference can go up to 6-7%. As I said, we can translate those water retention curves into equivalent pore radius distributions. And if we start here with the conventional plowing, you see very nicely that we create some big pores. Yes, these are some big pores after tillage. We stay active in the mid-season point, and then it seems that they are reduced in until the summer, let's say, until the fall. However, the most important for me, or a little bit something that was not expected so much, was that it seems that these differences are also part in this, let's say, medium range, where this is the pore size between field capacity and wilting point. This is the plant available water. So you see that we have temporal changes also of these pore spaces, and we have more or less from the mid-season to after harvest and after tillage, we have creation of more of these, should I call them mesopores? I never concluded on the definitions, but you see that we have more creation of those pores. If we check now the soil hydraulic properties and the hydraulic conductivity curve, what you see here again is the direct seeding data and the conventional plowing, and you see 3 types of data. We have here at PF1 the K-SAT value, the saturated hydraulic conductivity. This cloud of data here is the unsaturated hydraulic conductivity measured by the MSF, and this is the typical data from the high probe. And what you see also very, very nicely is that when you see the variability of the data, independent of the time, but just compare the variability, you see that we start with 5 orders of magnitude here in the saturated hydraulic conductivity, then we drop to 2 to 3 orders of magnitude in the MSF data, and then we drop to 1 order of magnitude here at the dry range. And we are still investigating now what is happening, and you see some difference. For example, you see that the mid-season here data measured by the MSF are lower than the after harvest and after tillage. We are still investigating why this is happening, but I just wanted to show here with these lines that even for this type of data here, if we compare the middle of its data cloud, we can see that we have a difference of around 10 to the 0.2, 10 to the 0.2, 10 to the 0.3, which is a factor of 1.7 times 2 of the conductivity. So again, we need time to interpret all this data. We have even more 3 points, and we have many, many data. So we try to interpret that, but for me, I think it's obvious, and that's something also that was one of my questions here, is that soil hydraulic properties change at the temporal, at the seasonal scale. So this is quite important. And then the question is how we can model that. So how we can model those soil hydraulic properties changes, and I think Nick talked about the USSF model, but I want also to highlight that there is another model developed by UFZ Halle. It's called the Bodium models. They are quite different. One is based on the Richards equation, one I'm not sure. But what we did, actually, we worked a little bit with the USSF model, and we focused a little bit on this relationship between soil structure and soil water flow and storage, actually soil structure and soil hydraulic properties. And here we did some extensions for the in this part of the USSF model. So we kept all the biology, all the drivers are kept identical to what Nick presented, but we changed a little bit some of the soil hydraulic properties. First of all, we didn't use the Brooks and Corey model. We used a more physically based model here for the dry range of the water retention curve and of the conductivity curve. We use the Brunswick model, but there are plenty of models that someone can use for that. But here we did that because we wanted a more realistic, a more physically description of this dry end of the soil hydraulic properties. And we didn't do that because we just wanted to deviate from what Nick did, but there are very strong evidence that the way we parameterize the soil hydraulic properties in this dry range affect a lot the calculation of the actual evapotranspiration, including actual evaporation and actual transpiration. That's why we did that. This is just some results from a recent paper where we actually investigate this exact point. We have those data and of the retention curve and on the conductivity curve, and we fit either one time with the blue point here with the typical Wagenknecht-Moale model, or we fit that data with this extended model that has a more, let's say, physically correct representation here of the dry range. And then we say, okay, what is the effect on cumulative evaporation and what is the effect on cumulative transpiration? And I'm not going into the details here, but I want to make sure that it's obvious now that just this difference on the dry range affect a lot cumulative evaporation and cumulative transpiration. So that's why we insisted to change this model. And then what we did, we extended also the model in the structural pore, let's say, size. And here we went a little bit more mechanistic, and we can discuss if this is a good approach. I'm not always believing that the more detailed or the more complicated is better. So I'm not like that, but I hope I can explain to you later why we did it in that way. But anyway, so this is the fracture model of Tuller and Orr. We have a unit cell here of the macro porosity, and this can be very flexible, let's say, based with this model, unit model. We can describe different kind of structural pore sizes and shapes. And you can think of it that with this model, we can have the same, let's say, structural pore size distribution, but we can be flexible and have different structural surface area of the structural pores. And this is actually, I'm going to talk about that a little bit later. And the whole model now is described by a matrix pore size distribution and a macro pore or a structural porosity size distribution. And by those two, we can construct the soil hydraulic properties. So another thing that we added actually in the model is that only for the structural pore size distribution, we are describe them with the Fokker-Planck equation. I will explain in a minute what does it mean. But first of all, based on this equation here, we can calculate a structural pore size distribution, and then by just plug in the Tuller and Orr model, we can calculate the soil hydraulic properties of the structural pore size. So, or if you prefer, of the macro porosity, we have the soil hydraulic properties of the structural porosity, and then by using this Fokker-Planck equation, we can calculate the gradation of the structural pore sizes. For example, if this is our initial distribution, a process changed that, and now we have less of those pores. We can directly see how the properties change, but we can also have drift, like shift to the distribution towards smaller pores, and this is again translated to soil hydraulic properties. Or we can have also change in the width of this distribution, and this is called dispersion. And then again, we calculate the effect on the soil hydraulic properties. In reality, we will have something that change. In reality, we have a degradation, drift, and dispersion. Now, you may are wondering, do we need some mechanistic model? And here, that's why we call that also a framework, is because our idea is that maybe we can, maybe not now, but maybe in the future, we will be able to construct one of those equations for each process. Let's say earthworm activity. We can come up with one Fokker equation for earthworm activity, one Fokker-Planck equation for, I don't know, for another kind of driver. And then we can offer more or less a framework that when someone knows how this distribution changes, to parameterize that in the model. If this is going to happen, we have to wait, I think. Okay, to test the model, we just took the daisy soil crop model and we simulated two agricultural systems, a conventional tillage and conservation agriculture. We have almost the same rotation. The only difference is that here in the year 2, we have a catch crop. And the main difference is that in conventional tillage, we plow before we sow the main crop. But in the conservation agriculture, we don't plow. We don't plow any tillage operation. And we run the simulation for 40 years, from 1951 to 1991. For 20 years, we had conventional tillage. And in 1971, we switched to conservation agriculture. If you see here, I wrote no feedbacks because there is a very important why we did that. Actually, this is a weak coupling. We run the DAISY model. We extract all the states that we need to run the USSF model, and then we calculate the results. In reality, there is no feedback between the modified USSF model and the DAISY model. That's something that we are going to do in the next years. Okay, and here you see some of our results. For example, we have the macro porosity for these 40 years, and you see that this period until 1971 is the conventional tillage. For example, you see very nicely that we plow the soil, and then we have maximum macro porosity, and then consolidation take place, and macro porosity is reduced until the next year, where we still have the next tillage operation. And if we compare the topsoil with the subsoil, you see that the more we are on the topsoil where the plowing takes place, we have bigger differences, but here in the subsoil, we don't have almost no differences. And then once we switch from conventional tillage to conservation agriculture, you see that biology takes over and we have an increase of the macro porosity, which become more stable. So after 15 years, we have an increase of 4%. At least 4%. And you see also some seasonal dynamics here, which are coming from the swelling and shrinkage of the clay minerals. So we can see here what's happening in the matrix, and you see very nicely that once we switch from conservation tillage to conservation agriculture, we have a reduction in the matrix porosity, and that's something that we actually measure in some of our long trials, that when we have conservation agriculture has higher macro porosity, but the matrix is more compacted. Actually, that's what we simulate here. And the same you see here with the saturated hydraulic conductivity. I just use that because everybody is familiar and we can relate with this huge actually increase from 3 orders of magnitude, from close to 1 to close to 2,000. And then once we switch to CA, you see that we have a stable increase following this kind of macro porosity. So another thing that we can do with this model, we can also see in a calendar year from January to January how the KSAT is changing for the two systems. Again, we have on the left conventional tillage, conservation agriculture, and here with the blue line, we have the net precipitation, and you see that the two systems had totally different dynamics, mainly because of the plowing. So here you have 3 peaks, 1, 2, 3 peaks, and this has to do, it's the dates that we apply in the plowing operation, but you see very nicely that we apply in the plowing and then it takes some months from the consolidation to happen. and actually it happens over the whole winter. However, in the conservation agriculture, you see that it's mainly the swelling and shrinkage that drives these changes actually, and we have more increase, let's say, of the macro porosity, or during the summer where we have the formation of the cracks due to the shrinkage. Okay, complicated model, yes, but the reason why we did that is because what we can do with this kind of detailed geometric models is that we can simulate some other states that are useful for other processes. I will give you an example here on the left. We have structural pore size distribution and probability density function. This is the blue line is before applying the plowing. You can think of it, we have bigger pores, yes. After applying the plowing, excuse me, so we have bigger aggregates, bigger structural pores, and this is before applying the plowing. But by assuming this kind of geometry at the smaller scale, we can also calculate the distances from matrix point to nearest pore surface, and we can also have a state. We can calculate other states like surface density distribution at each day. So we can do the simulation at each day, at each season, and we can also have some kind of how the average of these distances. And if you think about those distances, it's nothing more, or those distribution is nothing more like how aggregated the soil is, how big the aggregates is. So if you remember my what I said before about biological activity and how this is related with the distances from matrix to nearest pore surface. So this, in my opinion, will be, it's very attractive because we may be able to close the loop again. So soil structure, distances changes, and then back to biological activity. But again, this is to be shown. So we are still not so far. So to my conclusion, there is clear evidence of seasonal scale, temporal variability in soil hydraulic properties. And these changes affect the full range of soil hydraulic properties, or at least the wet range until PF3, PL4. Soil hydraulic properties remain useful description. I think we should measure soil hydraulic properties, but it seems that they should be treated as states to understand the changes that are taking place in the system, and not like intrinsic soil properties. And as you see that we have now some new generation models that can present these temporal soil hydraulic properties. However, again, for the modeling, the seasonal changes requires further development. I think, in my opinion, more research is needed to identify which processes are influenced by these dynamics. From whatever I have seen until now, I would guess, if I had to guess, I would say these changes are more important when we are talking about preferential water flow, transport of chemicals, rather than evapotranspiration, but we are going to do that systematically. And I think it's very attractive to introduce additional state variables so we can link soil structure with reactivity or microbial activity or organic matter dynamics. And I will close with one question on my presentation. I think there is always this question we can always increase mechanistic model. However, how mechanistic is useful, maybe that's something we can discuss now.